Thanks, Constantine, for the introduction, and thanks to the committee for selecting me for this talk. So today I'm going to be talking about a novel regulator of GCase and how CLCN3 regulates lysosomal function in Parkinson's disease. So first, Parkinson's disease is characterized by the loss of dopaminergic neurons in the substantia nigra. And in these neurons, we see the accumulation of aggregated synuclein, as well as the eventual formation of Lewy bodies. Normally, the lysosomal autophagy system can help prevent the accumulation of this misfolded synuclein. But when we see dysfunction of this lysosome, we also see the accumulation of toxic oligomers and eventually the formation of Lewy bodies. As you all know, Parkinson's disease is largely a sporadic disease where GBA is the most common genetic risk factor. But even patients without GBA still show reduction in GCase activity. So we asked ourselves the question, could modulators of GCase be hiding in the GWAS? So what I'm showing you here is the most recent GP2 genome-wide association study. You can see there's over 130 different loci, and the question of where to start looking and which genes to focus on is actually quite a complex problem. But thankfully, the Ganor lab produced a machine learning algorithm which incorporated a bunch of genetic and expression data in order to prioritize which genes were most likely to be most associated with Parkinson's disease in each locus. And from that analysis, you can see that CLCN3 was actually given the highest predicted probability out of any gene across the entire GWAS. It is found in locus 43, and you can see that a majority of the SNPs all fall within the introns of CLCN3. But more importantly, when you look at the expression data and eQTLs, SNPs that are associated with a reduced risk of Parkinson's are actually associated with increased expression of CLCN3, while SNPs that increase your risk of Parkinson's disease actually lead to reduced expression of CLCN3. So with that data, we were confident moving on to produce a knockout in iPSC-derived dopaminergic neurons, as you can see here. And the first thing we looked at was GCase activity. And sure enough, using the LysoFQ fluorescent probe, we see a 70% reduction in GCase activity when you knock out CLCN3. But why does this happen, and what is CLCN3? Well, unfortunately, there are no papers linking it functionally to Parkinson's disease. But from the literature, we do know that it is a transmembrane protein expected to be on endolysosomal membranes. And so that was the first thing we confirmed in our dopaminergic neurons. We used an antibody to co-stain with LAMP1, where we see strong colocalization. So it is on the lysosome. From there, the expected function is that it is a proton-chloride exchanger that helps facilitate acidification of the lysosome. But how does this work? Well, normally, the VATPase uses ATP to pump protons against both the electrical and chemical gradient in order to acidify the lumen of the lysosome. And what CLCN3 does is it exchanges one proton for two chlorides, bringing in two negative charges that helps reduce the electrical gradient and make the ATPase work more efficiently. So based on this expected function, the first thing we wanted to look at was the acidity of the lysosome in our neurons. And we did that using pH slice screen, which is a pH-sensitive probe that fluoresces more at more acidic conditions. And as you can see, that fluorescence is reduced when you knock out CLCN3, indicating that the acidification is indeed impaired in our knockout neurons. Next, we wanted to find out if this change in pH also affected proteolytic activity. And while using DQBSA, which is a probe that fluoresces more with more enzymatic activity, we see a reduction in fluorescence when you knock out CLCN3. So the pH is in fact impairing activity. So overall, we're seeing a reduction in acidification, a reduction in proteolytic activity, and a very strong reduction in GCase activity. But why do we only see a 10-15% reduction in overall activity for all the enzymes, but we see a 70% reduction for GCase activity? Well, to answer that, we moved to a simpler cell model, which is RP1, which is a cancer cell line, and produced a CLCN3 knockdown, where we were able to show that, again, the acidification and proteolytic activity were reduced with a strong reduction in GCase activity. So with this model, we decided to take a closer look at overall protein levels of a number of different genes, starting with CLCN3, which was knocked down successfully, and then, of course, GCase. And here we do see a slight reduction in overall protein levels of GBA, which could be contributing to the overall deficits in activity. The next thing we plotted was cathepsin B. And so this is another major Parkinson's disease risk gene. It's a protease that has an immature form, which is this top band, and a mature, more active form, which is this bottom band. And as you can see, when you knock down CLCN3, we seem to affect the processing of this gene, with the accumulation of the immature form and almost a loss of the mature active form. And so why is this interesting? Well, previous work in our lab showed that when you knock down CTSB, you see a reduction in GCase activity. And this is because cathepsin B is expected to cleave prosupposin into supposin C, which is a co-activator of GCase. So what do we see when we look at this in our cell lines? Well, when we plot for PSAP, we actually see an increase in prosupposin and a reduction in supposin C. So what we think could be happening here is that the pH is affecting not only GCase, but also cathepsin B activity, which is reducing the levels of supposin C and is unable to activate GCase. So this, along with the reduction in protein and the suboptimal pH, could explain the drastic drop in GCase activity that we're seeing. So clearly, when we impair CLC3, we see lysosomal dysfunction. But do we also see this change and accumulation of alpha-synuclein? Well, we turned back to our neurons and we did a stain for synuclein using an antibody, alongside a TH antibody, which is a marker for dopaminergic neurons, and a MAP2 antibody, which is a marker for general neurons. And if we zoom in on this one cell, we see that it is a dopaminergic neuron, as it's positive for TH. And when we look specifically at the synuclein staining, we see a drastic increase in the intensity when we knock out CLCN3. So this suggests that the endogenous levels of synuclein are increased when you knock out CLCN3, which is something we confirmed by Western blot. And it led to the question of how would these neurons deal with aggregated forms of synuclein. And to test that, we use PFFs, or preformed fibrils of synuclein. So this is a synthetically aggregated form of synuclein that we treat our neurons with for 2 weeks before staining for phosphosynuclein, which is a marker of de novo aggregated synuclein that kind of tracks the pathology of Parkinson's. And so as you can see, when we quantify this, we see almost double the amount of phosphosynuclein inclusions in our CLCN3 knockout neurons compared to our wild-type controls. And you can see that clearly here with these green spots all throughout the dendrites and cell bodies of these cells compared to the wild-type. So clearly these cells are less able to deal with this pathological synuclein. So overall, what we think is happening is that CLCN3 in Parkinson's disease has disrupted exchange of protons and chloride. This leads to reduced activity of the VATPase and an increased pH in the lumen of the lysosome. And with the suboptimal pH, we see deficits in a number of different enzymes, including cathepsin B and specifically GCase, which together lead to the accumulation of alpha-synuclein and eventually toxic effects that we see in the brains of patients. But a key takeaway from this is, again, that GCase is never working in a vacuum. You always have to consider other things like genetic interactors and especially the environment that it's in when you're studying its activity. And so with that, I'd like to thank everyone in the Fon and McPherson labs, specifically Nathan and Roxanne for their help on this project, as well as collaborators at the EDDU who generated the iPSC knockout line, Worral lab for help prioritizing the genes, and then Constantine actually for most of the genetic analysis that you saw in the presentation today. And thank you all.